Hello learner, if you are a biology student, welcome back to our lessons once again. I am giving you content and today we are doing something very interesting, and that is food test. Remember, in biology we have three papers. We have paper 1, we have paper 2, we have paper 3. And now in paper 3, this one is a practical paper. So I'm interested in sharpening you and ensuring that at least when you go for paper 3 in biology, you are able to answer these questions in a good way. We have a very common question that is from food test. Remember in Form 1, when you were talking about chemical compounds of life, you talked about certain foods. And I can just briefly remind you that we have starch, we have reducing sugars, we have non-reducing sugars, we have proteins, we have lipids. Lipids can be fats, can also be oils. We also have vitamin C. Vitamin C, that is commonly known as ascorbic acid. Ascorbic acid. The essence of studying these things is to understand. For example, if your food sample has these ones, and also to enable you understand the importance of these food substances in human body and how it helps animals to get good health. Now, our interest today is to discuss how you do what we call chemical tests. Chemical tests. How you check whether a food substance has got the following, or a food sample has the following foods. If you talk about starch, we have a chemical that we shall use called iodine. If you talk about reducing sugars and non-reducing sugars, we have Benedict's solution. And of course, we have several others, especially in non-reducing sugars. If it is proteins, we have sodium hydroxide, we have copper 2 sulfates. We also have sodium hydroxide solutions. All these are solutions. In lipids, we shall have a physical test. I will be able to explain that. And in vitamin C, we have DCPIP. This one is dichloro. I want to write it in full. Dichlorofenolindophenol. Yes, it's a very long name. And if you're not able to remember it, you can simply write DCPIP. I hope you will read and understand the importance of each and every food and how its deficiency in your body, you will suffer from some given conditions. So sit back and let us start with starch. So the first food, let us start with starch. Starch. Now, from the types of foods, from chemical compounds of life, by chemistry that you did in Form 1, starch is a type of carbohydrates. We call it polysaccharide. It is a complex carbohydrates that is stored in plant tissues. To your attention, remember we also have the carbohydrates that is stored in animal tissues. This one is called glycogen. So glycogen is for animals, starch is for plants. Both of them are carbohydrates. This is a complex carbohydrate we call polysaccharides. Chemical that we use in testing for starch. Chemical that we use in testing for starch. So starch is a food substance. So the food test here, food test, is called starch test. Starch test. When you are asked about the food test, look at that table properly. You will be saying starch test. We also have chemical tests. Chemical tests. If you are asked to state the chemical test, there is a difference. You say the chemical reagent, that is iodine tests. This is how you cite the difference between food and chemical tests. The reagent that we use, chemical reagents. Chemical reagent is one. Chemical reagent is iodine solution. Iodine solution. Please don't write iodine without solution, because iodine can be in crystal form. And when it is in crystal form, you cannot be able to understand or even be able to test for starch. So iodine solution, which is brown in color. Brown in color. There is an importance of understanding the color of that solution. Now we go straight to a typical procedure, or what you are supposed to do when you are testing for starch in a given food sample. So procedure. Procedure. So if I talk about the procedure, I want to assume I'm using 2 ml, 2 centimeter cubic of the food substance. So you say put, for example, 2 ml or 2 centimeters cubic of food substance, the food substance that you are checking, into a clean, dry test tube. Then add 1 ml of iodine solution, and shake. Observe and record. Observe and record your observation. This is a typical procedure. In any question in KCSE, Biology Paper 3, you will be required to write the procedure on your own. You are not given the procedure. This is not like chemistry where you're given the procedure of what you're going to do. Biology, you write your own procedure. So from there, if the starch is present, the color changes will be as follows. So we go to observation. This is for a positive reaction. If the starch is present in this food substance, then this is what you will observe. Color changes from the background color of iodine, which is brown. So color changes from brown to black. You can get either black, you can get blue, you can get dark blue. There are so many students who say blue-black. Blue-black. There is no color called blue-black. I'm cautioning you. We don't have a color called blue-black. Please say you get black or blue or dark blue. Avoid saying blue-black. Then that means in the conclusion, starch present. Conclusion. In the conclusion, you say starch present. Do not say starch is present. Just say starch present. Don't say starch. Just say starch present. That is all about starch. Remember the color of the chemical reagent, brown, which is going to be the background color. Background color. And then now the color that is formed after the reaction is positive. That is all about starch. We go to reducing sugars. Reducing sugars. Food number 2, we have 6 of them, so we talk about reducing sugars. Reducing sugars. The category of these foods actually reducing sugars are monosaccharides. They are monosaccharides. And that means we call them simple sugars. Simple sugars. So, you should know the examples of these sugars. Actually, we have the most common one is glucose. We also have another one called fructose. Then we have the third category called galactose. All these 3 are reducing sugars. They are simple sugars. They have almost the same characteristics. In fact, if you taste them, they have they form sweet tasting solutions. You may not even be able to differentiate between them, but as long as you understand that they are reducing sugars, that one is good. Now, the procedure for this, but before that, we have the chemical reagent for this. Food test, reducing sugars test. Chemical test, Benedict's test. I think we are together there. Because the chemical reagent is Benedict's solution. Benedict's, the correct spellings here. There must be an apostrophe between T and S. Benedict's solution. And the color of Benedict's solution for the background color thing is blue. So you must always understand the color of the reagent, which gives us the background color. Now from there, we go to the procedure. The procedure for testing reducing sugars. And I'll tell you why they are called reducing. There's a reason for that. Procedure. I think we shall still use our 2 ml. We put 2 ml of the food substance into a clean, dry test tube. Then you add equal amount. So 2 ml. I want to stress, I want to write it in capital letters. Equal amount of Benedict's solution and shake. Then the third step, boil the mixture. Boil the mixture for 3 minutes, or you can say 4 minutes, and at least or at most 5 minutes. There is a reason here. Boiling must be quantified. The amount of time you take in boiling must be quantified. Some people say boil, just boil. Others will say heat to boil, which is okay. When it is heat to boil, you will heat it until when it has boiled, you now finalize your process. As you boil, observe the color changes. And record. Before I get out of the procedure here, I will take you back to the amount of chemical reagent. It is always good to indicate how much chemical reagent you're using. Same way, when we were doing starch, we gave you, I told you 1 ml, half. There are only 2 chemicals. When you're using them, you use equal amount. The first one is Benedict solution, it is here. The other one is sodium hydroxide that is used in testing for proteins. When we reach proteins, I will tell you we shall use equal amounts of the chemical reagent sodium hydroxide and Benedict solution whenever we are using them with that of the food substance. What happens if the reducing sugars are present? Check on the observation for a positive reaction, and that is if reducing sugars are present. This is what you will see. this is what you will see. For a positive observation, observation, color changes from, you start from the background color. When you mix the food substance with the Benedict solution, the original color or the background color is blue. Color changes from blue. Then we have a sequence of colors. 2, we have blue, 2, green, 2, yellow, 2, ROB. This is not one color. This is a collection of 3 colors. I will tell you, R stands for red, O stands for orange, and B stands for brown. So in the final color can be any of the 3 colors, but most people get orange or sometimes red. The sequence of color changes. You start from the background color, you go to green, you go to yellow, and then one of these. Please be very accurate. Now, there is an indication here. If there is no color change, if the blue color persists, blue color becomes persistent. If it persists, if the blue color persists, then no reducing sugars. Non-reducing sugars are absent. If the blue color of the solution remains or is unchanged, then it means there are no reducing sugars. If you get the final color as green, it means very little amount of reducing sugars are present. If your final color is yellow, then it means average amount of reducing sugars are present. And finally, if your final color is ROB, any of the 3, then it means we have high amount of reducing sugars. But just be very keen. This one is not what you asked. You just asked to test whether they are present. Whether they are little, whether they are average or high amount of reducing sugars, we are not interested in the amount of reducing sugars. Our interest is, if you see the color changes to that end, you say reducing sugars present. Conclusion, reducing sugars present. That is all about reducing sugars. Number 3, non-reducing sugars. Non-reducing sugars. Non-reducing sugars, number 3. Non-reducing sugars. These ones are complex sugars. They are called complex sugars. That is why we call them disaccharides. Because they are formed as a result of condensation of monosaccharides. If you bring 2 glucose molecules, condense them together, glucose plus glucose, you get maltose. Maltose, of course, plus water. I'm taking you back just to remind you a few of these things. If you have glucose plus fructose, you form sucrose plus water. And finally, if you have glucose plus galactose, you form what we call lactose plus water. What I mean is, these ones are monosaccharides that have been condensed to produce. So these 3 are disaccharides. They are examples of non-reducing sugars. So now, chemical reagents In this one, we have several. We don't have just one. We have several chemical reagents, and the procedure is slightly longer than that one of reducing sugars. Chemical reagents. Chemical reagent or reagents. The first one is Benedict's solution. We have another one called dilute hydrochloric acid. We have another one called sodium hydrogen carbonate. Sodium hydrogen carbonate solution. Sodium hydrogen carbonate solution. I think those are the three substance or chemical reagents that we shall be using. As I take through my notes to ensure that we are accurate on that. Very nice. You must say sodium hydrogen carbonate solution. If you don't say solution, then you get it wrong. The procedure now. The procedure. Procedure for testing for non-reducing sugars. Procedure. Procedure for testing for non-reducing sugars. Put 2 ml of the food substance. Put 2 ml of the food substance into a clean, dry test tube. Into a clean, dry test tube. Then add 1 ml. Add 1 ml of dilute hydrochloric acid. Add 1 ml of dilute hydrochloric acid and shake. Shaking is important because you have to mix the food uniformly with that chemical reagent. So shaking is always very, very important. Then boil the mixture. Look at the amount. I have used 2 ml of the food substance. I have used half of the chemical reagent, dilute hydrochloric acid. So boil the mixture for 3 minutes. 3 minutes. This is my decision to boil for 3 minutes. Otherwise, you can have 4 minutes and at most 5 minutes. You can also say heat the mixture, heat to boil. Then after boiling, cool the mixture. Cool the mixture in a cold water bath. In a cold water bath. I hope there are several methods of cooling the mixture. You can open the tap, cold water, and then as the water is running, you cool that mixture. But mostly we talk about a cold water bath. Then after that, you neutralize the acid. And drop wisely. Actually, we need to quantify the amount of sodium hydrogen carbonate here. We are now at hydrolysis of or neutralization of the acid. Drop wisely. Add sodium hydrogen carbonate. Add sodium hydrogen carbonate solution until fizzing stops. What you are doing is sodium hydrogen carbonate is neutralizing the acid. Yeah, this is a neutralization process. You are neutralizing the acid so that at least that acid is complete. So when fizzing stops, then the air bubbles have completely been produced, because in the reaction of sodium hydrogen carbonate solution with HCl, this is what we get. We can talk about sodium hydrogen carbonate solution plus HCl. HCl, this is what you produce. You produce sodium chloride, sodium chloride. Then you produce carbon 4 oxide gas, carbon 4 oxide gas plus water. So in this case, you have fully neutralized. It is a balanced chemical equation. So you make sure that the acid is completely neutralized. What happens? After that now, you add another reagent. You go to the next procedure. And clean this one here. So you add, now we are at Benedict solution. You add 2 ml. This is equal amount of Benedict solution to that of the food substance. We said when you're using Benedict solution, so add 2 ml of Benedict's solution to the mixture and boil again as you observe. Boil again as you observe and record your observation. Boil again this mixture now, and as you boil, you record your observation. So if the non-reducing sugars are present, this is going to be the conclusion. Or sorry, the observation first. The observation made is going to be similar to that of the reducing sugars test, because first of all, we have broken down disaccharides into monosaccharides by dilute hydrochloric acid. We have neutralized the acid by a sodium hydrogen carbonate solution. And now we've added Benedict solution and boiled the mixture again to see the color changes. That means that process has completely given us reducing sugars, and so color changes from blue to green to yellow to either or any of the 3 colors I told you, R, O, B. It could be red, orange, or even brown. But again, you just write the one that you will find there. And in the conclusion, now that this procedure looks like the reducing sugars, we don't say reducing sugars, we say non-reducing sugars present. Non-reducing sugars present. So that is all about reducing sugar, non-reducing sugars. We go to proteins. Proteins, number 4. Proteins. Proteins. In proteins, we have 2 chemical reagents, and those ones are 10% sodium hydroxide, 10% sodium hydroxide solution. We also have 1% copper 2 sulfate, again solution. Sodium hydroxide solution is colorless. It's always good to understand the colors of these reagents. But copper 2 sulfate is blue. So we have 3 chemicals that are blue. Benedict solution, copper 2 sulfate, and finally we have another one, the DCPIP, that we are coming next, is also blue in color. The reason we understand the background colors is because of the color of the solution or the mixture before we start the reaction or before we start the experiment. So procedure, Procedure. Put 2 ml of the food substance into a clean, dry test tube. Then you must know the first reagent to use. Add equal amount. So equal amount here means 2 ml of sodium hydroxide of 10%. But now, it depends whether it has been indicated in the question or not. If it has not been indicated, please don't indicate these percentages if they are not indicated. Add equal amount of sodium hydroxide solution into that test tube, and of course shake. As you shake, after shaking, you add again into the mixture. Now this is copper 2 sulfate. Add 1 ml of copper 2 sulfate and shake again. Shake again as you observe the color changes, or as you observe the changes. So the observation, if proteins are present, observation, color changes from the background color, that is of copper 2 sulfate, blue, to purple. Checking the spellings of purple. To purple. Blue to purple. And then that means in the conclusion, proteins present. That is all about the proteins. We go to vitamin C, our second last. Vitamin C is ascorbic acid, number 5. Ascorbic acid or vitamin C. Vitamin C. Ascorbic acid, number 5. Vitamin C, which is commonly known as ascorbic acid. This one is found in fruits, and these vitamins are very important in the body because insufficiency, if these vitamins are not sufficient in your diet, then you suffer from what you call scurvy. Scurvy is a condition where we have bleeding gums. So these are conditions that will affect your teeth and jaws. So now vitamin C, chemical reagent is DCPIP. Yeah? If you know the spellings of all the letters of this word, dichloro, please write it. Dichlorofenol. So if you know this one, you write. If you don't know it, please stick to the capital letters of these editions. DCPIP. The procedure is slightly different. The color of this one is blue. It is blue in color. It's blue. Procedure. This time round, we are not beginning with the food substance. This time round, we are beginning with the chemical reagents. Put 2 ml of DCPIP into a clean, dry test tube. So this time around we are beginning with the chemical reagent, unlike the rest of the foods. After that, drop wisely, add the food food substance into the test tube. Of course, dropwise add the food substance. That is what you do. The quantity that you will add depends on how the reaction takes place. If if vitamin C are present, this is going to be the observation. Observation, very brief. DCPIP decolorizes. Decolorizes. In short, the blue color of DCPIP disappears. Disappears. What does it mean? If this color changes from blue to colorless, then you conclude that you conclude that vitamin C, of course, present. But if the blue color of the mixture remains, or of the DCPIP persists, then it means vitamin C are present in that food sample. Let us go to the last one. The last one is lipids. Lipids. Very last in this video, lipids. Yeah, lipids. Number 6. Lipids. Lipids can be fats, can also be oils. And now, we don't have chemical tests for this. There was a chemical test whereby ethanol was being used, but I think they abolished it because of the misuse of ethanol by students and maybe the examinations, and of course, it was a bit difficult and hectic to provide that ethanol to all the students who are sitting for an exam in this part. So we go to what we call a physical test. So no chemical tests. We talk about the physical test of lipids. Physical test, we call it a grease spot. Grease spot. The food that you are testing, whether it has lipids or fats, that food can either be in solid form or solution form. So that will dictate how I'm going to write the procedure for the test of lipids. So procedure. Spread. I'm assuming mine is in solution form. And most of the time they bring that food substance in solution form. So what you do, you spread. In this one, we don't have a test tubes here. We have no test tubes here. There is no boiling, nothing. So you spread a drop of the food sample solution. You spread a drop of the food sample solution on a filter paper. So if you have a filter paper like this, you put that drop here and spread it. Make sure it spreads a little, so that you can have a spot that you'll be looking at. Then hold, but if it was a solid, you will say rub. If it was a solid, you say rub. Rub on a filter paper that substance that you are testing whether it has lipids. Hold the paper, hold the filter paper against a source of heat. But you must be carefully, so as not to burn the paper, So what you're doing is, you want this spot to dry. So you want the spot to dry. That's why you're holding it against a source of heat. Maybe it could be a candle or some Bunsen burner, some flame or something, but you must be careful not to burn it, just to dry that spot. When that spot dries, you do the last procedure. You hold the filter paper with a spot, of course it is now dry, against a source of light. Mark the difference. The first one, you hold it against the source of heat, but carefully not to burn it, to make it dry. The next one and the last one is you hold it against a source of light, so that you can make an observation and see whether there is light passing through. What will be the observation if the lipids are present? Observation. If the lipids are present, this is what you will see. If the lipids are present, now the observation, a permanent, translucent spot is formed. A translucent spot is a spot that allows light to pass through it, but you cannot see through it. So if it will be formed and it is permanent, you cannot reverse it, then the conclusion is lipids present. Make sure you don't write lipid. That is a physical test for these lipids. So I think we are through with the discussion of the 6 food substances, but I want to remind you a few things. Time for boiling now. Generally, time for boiling. You must quantify the time. 3 minutes, 5 minutes or something. Then number 2, the amount of food substance. Then of course we also have something else you must quantify. Then the color, background color must always be marked, so that at least before you say a change has occurred, you must say it has occurred from a given point or from a given color to another color. Now that is the end of food test, and I hope you have done your best. Continue watching these videos, and until the next lesson, see you and have good time.